refractive index and is mismatch and the optical aberrations. So what are optical aberrations? This would be so it's an artificial point spread function. This comes from the Kurt Thorne, a very nice microscopy blog, which unfortunately has stopped now. So we have a theoretical point spread function, but this is a good point spread function. You see how tight we shrank the little rugby man to its bare minimum, and then we have a very symmetric point spread function. The real point spread functions are never actually that symmetric. In general, they are more blurry on one side compared to the other. And this here could be a distorted aberration, point spread function. So this is spherical aberrations in this particular case. So optical aberrations means that we have a distorted point spread function. So we are losing resolution, basically, especially in Z, but also in XY, but especially in Z. So the consequences are very much in Z, but depending on the aberrations, it could be also other stuff. It could be center to periphery. So the focus could be different center to periphery. We could have the different colors that are not focused at the same Z level. All these are optical aberrations. They can come from your lens being damaged or your lens being of a very low quality. I mean, the objective being of a very low quality or being damaged or being dirty. All this can create optical aberrations. Now, the optical aberrations that come from refractive index mismatch. So what I mean by refractive index mismatch, again, the objective manufacturers, when they design their objectives, they expect the light to just the excitation light to just go straight from the objective lens, not straight, but even to go undisturbed from the objective lens into the immersion medium of the objective, into the cover slip, and into the medium. So clearly, the cover slip might not be the same refractive index as the medium, for example, if we have an air objective, but this is accounted for as long as we know that the cover slip is 170 micron thick. So all these things are calculated in. So here what we have is we have an objective. And this is an inverted microscope, so you can see it because the objectives are pointing upwards and they are under the sample. This is our sample. So on the objective lens, we have some immersion medium, and this is an oil objective, so we have an oil immersion medium here. And then we have a cover slip, and then we have our sample. And our sample is fluorescent beads in a vector shield. So vector shield is a mounting medium, and it has a refractive index of 1.44. And I remind you that this little n is the way refractive index are abbreviated. So n, the refractive index of vector shield 1.44, of glass 1.52, oil, same thing. And the lens here is also made of glass, so it's also the same. So here in this vector shield, we have 3 fluorescent beads, and one of them is stuck on the cover slip and the others are further away inside. If we look at the point spread function that is created by this bead, so the image of this bead, when we image it, this is what it looks like. So Z equals 0 means that the bead is stuck onto the cover slip. If now we are imaging this bead, which is 25 microns away, and 25 microns, by the way, is about 1 cell diameter, so it's not very big. So that would be, for example, imaging a fluorophore at the bottom of the cell and at the top of the cell. Okay, 25 microns away. If we look at the point spread function, it is highly distorted. If now we look at the third bead, which is inside the medium, at 50 microns inside, to 2 cell diameter away, you can say 2 to 3 cell diameter away, then we get an even more distorted point spread function, which is completely asymmetric. So this is what happens when we do not match the refractive index of our sample, our mounting medium, and the immersion medium of the objective. And we're going to see more details about this with Jinjiang in the refractive index mismatch workshop after this. So different point spread function, the point spread function changes with the depth when we have optical aberrations. And this means that we would require, even if we have on our objective a correction color that may help, we would require anyway different positions of the objective correction color to be able to image these 3 beads with the same point spread function. So it's not possible to image these 3 beads in the same way if we have a manual correction color. It will not be possible to correct for the aberrations due to the RI mismatch, the refraction index mismatch in the sample. And as you've seen, the distortion grows exponentially with the imaging depth. So as soon as when we are close to cover slip, there is no distortion. As soon as we are further inside the sample, and the light basically has to travel through the medium before it, the excitation light before it reaches the bead, and the emission light before traveling from the point to the cover slip, then we have distortions that grow exponentially with the imaging depth. And this can be corrected if you have an automated correction color. And of course, if your medium is homogeneous throughout the sample, then you could correct this with an automated correction color, but not with a manual one and not with a normal objective that has no ring. So what happens if we zoom in a little bit and try to figure out what's going on? So here we have a cell and we have a little fluorophore in here. It's not against the cover slip. It's a little bit away from the cover slip. A cell has, it's basically full of, except if you look at adipocytes, the cells are basically full of cytoplasm, and it has a refraction index the same as water, so 1.33. And then we have the cover slip, which is made of glass. And here we can put different, so we use different type of objectives. So here we will have different media. So let's have a look. We have refraction here, because here we have a medium with a certain refractive index, and here we have another one. So there is refraction, as we have seen before, at the interface. And the light is bent, okay? And we're going from a low to a high refractive index, so it's bent in this direction. And it does the same effect as what I've shown you before, the pencil in the glass full of water. And as I mentioned before, that there is a law that regulates how much the light is bent, and it's called Snell's law, and it's related to, so the angle is going to be related to the difference between the 2 refraction indices. So if we have one angle in here, we're going to have a different angle in here. And if here, this is a water objective now, so we have water on top of the objective. So we have water in the cell, glass, and then water. So what happens is that this angle here, when we have refraction again, when the light comes out of the cover slip and goes towards the objective, we're going to have the same angle here and here. Okay. And this is the way manufacturers correct for the cover slip. So even if the light goes through 3 different media with different refraction indices, because we have the same medium here and the same medium here, we have the same angle, and therefore we can correct for the fact that we have glass in between. So that is counted in the objective. The objective is designed to have a cover slip. And this is the reason why I was saying if you have a dipping lens that is designed not to have a cover slip, you should not put one. Otherwise, you will put distortions in your image. So this is the real distance between the bead and the cover slip if we would want to measure this Z measurement, distance. And this is the one that we're going to measure, and it is the same distance. So there's no, our measurement is correct. So we get the correct data when we have a cell, which is very much like water, and a water objective. What happens if you use an air objective instead? So what happens is that we have the same angle in here because we go from water to glass. And then afterwards, when the light goes to air, so from glass to air, we're going to have a different angle. So when we want to measure this distance that we have here, the distance is going to be compressed. So the object is going to be smaller. It will look smaller than it actually is. Okay, so we have a compressed in our Z stack, the object is going to look compressed. And what happens if we use an oil objective? I'm sure you can guess. So we have here a certain angle, and because this has the refraction index of glass, the same so it's the same as oil. So we have the same refraction index here and here. So the light goes straight, basically. It is not refracted. And so we are measuring a distance that is much bigger. And therefore, we have an elongated sample. And this is how, when we have a refractive index mismatch, we have a scale distortion. And that has especially a very strong effect on the Z measurements. And especially when the stuff that we're measuring is away from the cover slip. But if we're measuring in Z, one way or another, there's always a part that is away from the cover slip. So these are the consequences of having refraction index mismatch. We cannot accurately measure objects in Z. So if you want to know if the angle increases or decreases, it's quite easy if you follow the light path of the emission. When the refraction index increases, then the image of the sample also increases. So when we go from for example, from here, yeah, exactly. So when we go from a water sample to an oil objective, then the image of the object is bigger. We increase. And this is much worse when the object that we're measuring is away from the cover slip. This is the reason why I'm telling you all the time, put your sample on the cover slip. And I know that there are some cases when it's difficult, because if you do antigen retrieval, your sample might detach, but then you put it horizontal and there's a lot less risk that it detaches. And then if you treat, we will see all this in the sample preparation workshop. But when you are using RNA scope in a section and you put it on the slide and you put a thick level of mounting medium, a thick layer of mounting medium, and you image with the wrong objective, this is really difficult to analyze, because from one day to another, you're going to have more or less mounting medium, and this will influence the brightness and the shape and the contrast of your images. So again, this is the effect of a refractive index mismatch is also that it lowers the numerical aperture. So maybe you're using a 60x oil objective because you think that, oh, this one has a high numerical aperture, so it's going to be great resolution. But if your sample is made of water or is in water, then what you're doing actually is you are actually effectively, because of this change in angle, you're actually closing the numerical aperture. You're not using the full numerical aperture of the objective. So you're using the 60x oil because it has a high numerical aperture, but the consequence of your experimental design is that you have a lower numerical aperture than what you should have. So you would be much better off with a water objective, where you would have no scale distortion and you would use the full numerical aperture of the objective. Okay, so in here you can see what this is a water objective with a water sample, and in here the image of the sphere is displaced, but the measurements are correct. And that is because the thickness of the cover slip is accounted for in the design of the objective, whereas in here we get distortion, we have a smaller numerical aperture, and the object is elongated. The interesting thing is when the sample, the part of the sample that is stuck against the cover slip, there is no distortion. Okay, so when we are close to the cover slip, no problem. It's only as soon as we get a little bit away, and even 10 microns, you already see this effect. So there is distortion away from the cover slip, and we have here again, we have some distortion at the cover slip and the same distortion away, and therefore we have no distortion of the whole sample. And this assumes that you use the correct cover slips. So you have to use 170 micron cover slips, which are called 1.5, number 1.5. So the cover slip is part of the design of the objective, as well as the sample and the mounting medium. And of course, the immersion medium of the objective. All this is part of the design of the objective. So choose cover slips number 1.5. Make sure that you match the refraction indices of your sample and your objective. Otherwise, you may artificially lower the numerical aperture of your objective. And I remind you that the numerical aperture is a combination of the angle here and the refraction index of the immersion medium of the objective. It's we multiply them. So we end up with a smaller angle and therefore a lower effective numerical aperture. So we have aberrations if we have mismatch, so that's a water sample with an oil objective. We have aberrations, we have elongated volumes, and we have a reduced numerical aperture. And if we use a water sample with a water objective, we have no aberrations, correct volumes, and full numerical aperture. So use water objectives with water samples, and water samples that includes cells that are live on or in PBS on the cover slip. If they are fixed, then you have to follow the refractive index of the medium that you're using to mount. Use oil objectives with high refractive index only and with mounting media. So you can figure out, and you're going to have to find today the refractive index of your mounting medium. So this is an example of a dipping lens objective. So it is designed to have no cover slip. It should just dip into the medium. And this is the sample is on the slide, and we have an aqueous medium. So we should use a water objective, and we have covered it with a 1.5x lens. But in here, we have no cover slip, and you can see the image where it looks very nice. But if we now use a 40x water objective, which is designed to have almost the same numerical aperture, but it's designed to have a cover slip, you can see that the image is much sharper. And so clearly, because we have a lower contrast in these images, we definitely have a higher resolution when we have a higher contrast. So we'll see this next week. But you will only see the difference if you compare the images side by side. Otherwise, it's impossible to see that there is a problem with this image. It just looks a bit hazy, but you won't notice it. So not matching the objective and the sample refraction indices or using the wrong cover slip thickness leads to increased optical aberrations, reduced brightness, reduced resolution, loss of contrast, incorrect measurements in Z, and decreased effective numerical aperture of the objective. And that is a major cause of signal loss when imaging thick samples, because both the excitation light and the emission light are effective. Both of them have to go through the objective and through the mounting medium before they get to the sample. However, if the sample is extremely thin and stuck against the cover slip, it doesn't matter. So there you can get away with murder and use any objective and not match the refraction media. Doesn't matter, but this is only in the case of a sample stuck against the cover slip and only the part that is stuck against the cover slip. The aberrations appear just a few microns away from the cover slip, and I'm sure that you have seen that when you image a monolayer of cells on a cover slip. The bottom of the cell is always much sharper than the top of the cell. So it's not many microns between the bottom and the top, and we clearly already see the effect of refraction index mismatch. And the problem increases proportionally with depth. So you can count that a tissue will have more or less a refractive index of 1.4, and it's going to be a bit higher for fatty tissues. And then if you have cells on the cover slip or cells in a gel, then you go for the same refractive index as water, and you use a water objective. So how can we prevent optical aberration and refractive index mismatch? We use the correct cover slips, either 1.5 or 1.5H, which have an even better precision. We do not use cover slips with dipping objectives. They are designed to have no cover slips, so don't use them with cover slips. You put your sample on the cover slip directly, not on the slide. So you put your sample on the cover slip, you add the mounting medium, then you mount it with the slide. So your sample will look the same, but the sample will be against the cover slip. We'll see this in the sample preparation. video or lecture. Exactly. And always adjust the correction color of your objective if there is one, and match the sample and the objective refraction index as much as possible. If it is not possible for you to match, use an objective that has a lower refraction index than the sample. So this way you can use the full numerical aperture. If your objective is an oil objective with a water sample, you're not going to use the full numerical aperture. You will reduce artificially the numerical aperture. But if you have an air objective with a water sample, at least you're using the full numerical aperture of the objective. If the sample touches the glass and is very thin, any objective can be used. And tissues have a refractive index of about 1.44. So they are best imaged with a silicone or glycerol objectives, which have this the same refractive index, or they should be cleared. And we will talk about this in the clearing lecture. Live cell in medium are best imaged with water objectives. And here I have a little quiz for you. This is from a previous year student. Do you see any problem in this image? So it's imaged with a 10x objective, an air objective. The sample is on the slide, and then there's prolonged diamond, and we have these channels, and we have focused on the red channel. You see anything? It's difficult to see, right? So I show you like this, maybe you will see. Any issue?
And this is with which microscope?
I do not know. It was one of the students' microscope. I think it was the Zeiss confocal.
There's a problem of low resolution.
Low resolution? Where do you see that?
It looks like out of focus to me. But
the
Which one
blue
is
is out of
focus?
focus, but the green is.
Exactly. So we're focused. We focused on the red. We focused on the red, and the red is in focus. The green is in focus. The blue is not in focus. I've told you what it is, right?
Yeah, you said it's because of the refractive index mismatch.
This is because the sample is on the slide, and we have a refraction index of prolonged diamond, which on top of that was autofluorescent, but you couldn't see in these images. So we have and the sample is on the slide, and we have prolonged diamond, and we are going through, and we see this very clearly. The channels, the three channels, are not focused at the same plane. So there are chromatic aberrations. The blue channel is out of focus when we focus on the red one, and the mounting medium on top of that was autofluorescence because it was old, and so it's not visible in here. But this is something that, again, you need to keep a critical mind. When you see something like this, all your bells go out. Try to put as little mounting medium as possible. If it's not possible for you to put your sample on the cover slip, you should put very, very little mounting medium. Put your sample on the cover slip, not on the slide, and show each channel separately in black and white, because you see when I showed you the overlay, you didn't see it. It was very difficult to see, but now you could see this straight away. Good. So I want to see how I am late or what. I hope not. So I'm here. Oh, yes, I'm actually late. I'm sorry about that. I know I'm not late. I'm only 1 minute early. Good. So do you have any question about this? Yes? We had to raise our hand. Let me see. Yes?